Okay, so I spent quite a bit of time studying evolutionary shifts in flowers from one pollination syndrome to another. And I've been studying this group, Penstemons, for years. I have several collaborators here whose names are listed. What we found, in brief, is that over and over again in this phylogeny of these flowers, Penstemons, we've had shifts from bee-pollinated things, bee and wasp-pollinated things, to hummingbird-pollinated things. And we've never had shifts in the other direction. And these shifts have a kind of predictable quality about them. That is, several different characters tend to change at once when they have a shift like this. And that's happened not just in Penstemons, but in a number of other groups, although you always have to think about it in terms of the group that's under study, because the group also brings to the evolutionary dynamic its own characters, and its characters determine that evolutionary dynamic partially as well. So for instance, if you have flowers that are radially symmetrical, then the evolutionary dynamic would be a little bit different than if they're bilaterally symmetrical. And it also turns out that this dynamic is really strong in North America, and so far the people who have studied it in South America and Central America haven't found the same patterns. So location also matters. Now, before I really get into the adaptive nature of these flower traits, I wanted to liken them to sexual selection. And here we have the anther of one of my flowers. That's the structure that makes pollen and presents pollen to bees and to hummingbirds. And I think that anthers, those male parts of flowers, are kind of like antlers. They're kind of like the male parts of deer, in that both of them are shaped to have peculiarities and excesses because of advantages in mating that are not necessarily particularly good advantages for survival of anything, the individual or the species. Okay, now this brings up this theory that we've developed called pollen presentation theory, and it has a couple of tenets. One of them, the first one I'll call it, is the theory of diminishing returns, where if you have a flower and it presents different amounts of pollen in different imaginary scenarios, then the amount of that pollen that actually makes it onto other flowers' stigmas, so is successful at pollination, that is a decelerating curve. If it presents a little bit of pollen, then it gets this much to stigmas. If it presents twice as much pollen, it doesn't get twice as much of the pollen to stigmas. So what that means is that if there's lots of visitors visiting the flowers, there's a high traffic rate, then it's adaptively in the best interest of the flowers to present a little bit of pollen at a time to many visitors, each of which would then deposit a large proportion of that small amount that they removed. So I can go like this and say, here's the first little aliquot of pollen that's presented, and it's removed by an animal and then deposited on stigmas at that level. And then you have another anther that opens up and it presents more pollen, and it's visited by another animal, and that animal takes a large proportion of that small amount of pollen and deposits it on stigmas. And then you have another one, and another one, and another one. So by gradually presenting pollen, the flowers are, in essence, linearizing this relationship and getting more of their pollen out to stigmas and making it so it's more likely that they will sire seeds than a flower that just opened up all of its anthers at once to the pollinators. So that's the first idea of this pollen presentation theory. I'll state it again. If there's a single kind of pollinator and it's reliably abundant, so there's a high traffic rate, then dosing pollen gradually increases male fitness. Now we bring up the second tenet of pollen presentation theory, and that is that different kinds of animals, like bees and hummingbirds, different kinds of animals can differ in pollinator efficiency, that is, in their capacity to remove pollen and then deposit that pollen onto stigmas. When the better kind of a pollinator is abundant, the less efficient pollinator becomes a conditional parasite. So I think that in our case, the hummingbirds are the better kind of pollinator. The hummingbirds, they don't eat pollen. They don't groom that much. They just sort of let it get on their face. And they fly long distances between the flowers, relatively long distances between the flowers, whereas the bees are in the business of raising baby bees. And the way that you raise baby bee bodies is to feed them pollen. That's where they get their protein from. And so bees go up to flowers and they rub against the anthers. They get a lot of pollen on their body, and then they start grooming it into little places that are relatively unlikely to be touched by a stigma. They take it home and they feed it to their developing larvae. Then they also tend to sort of stay on one flower and do all of this rather than going boom, 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 and hitting a whole series of flowers. So in this context, we think that if we had different kinds of animals, like bees and hummingbirds, that they would differ in their pollinator efficiency. That is, hummingbirds would be more efficient and the bees would be less efficient. And then when the better kind is abundant, when the hummingbirds are abundant, the less efficient pollinator, the bees, would become parasites. So the first idea I had to kind of get you over was thinking that flowers are female and they're nice things that you put in your hair and stuff like that. No, really, flowers are male. They're like antlers. And think of deer charging at one another. Flowers are very masculine, very manly things. And then the other thing I had to get you over is thinking that bees are good for flowers. No, bees are only good for flowers if there's nothing else around. If there's hummingbirds around, then bees are bad for flowers. They're taking all the pollen, they're feeding it to their babies, they're not pollinating very much. And then all of this stuff is sort of true even if they don't differ in how diminishing the returns are. If they differ in the amount of pollen they deposit on stigmas as a proportion of what they remove. All right, so we come upon this great group of plants, which I'll call penstemons. It actually includes two genera, Penstemon and Kekiella. This is the group of flowers that has the most species that are endemic entirely to North America. And within this group, we find a number of subgroups, little groups within it. So for instance, there's this subgroup of Kekiella, Kekiella breviflora, and Kekiella ternata, where one is bee pollinated and the other one's hummingbird pollinated. And then we find some other penstemons. There's a little teeny group of penstemons that includes gentianoides and Hartwigii, and one is bee pollinated and one is hummingbird pollinated, and so on and so forth. So we can develop a number of different phylogenetic blocks, or little pairs of relatives that we can compare to one another. And we can compare them to one another in terms of what actually visits them, their color, the shape of the flowers, the amount of nectar they produce, the kind of nectar, and most importantly for us, the way in which those anthers open up and present pollen like antlers would to pollinators. From this, we develop these systematic rules, and here's sort of the way they run. So you might think of these as hypotheses that have been developed from classical literature, and then we're able to test these hypotheses with Penstemon. You could also go and test these hypotheses with other flowers that appeal to bees and hummingbirds in South America, and people have done that to a certain extent. And what we found is that the rules hold really well here in North America, and the rules hold much less well in South America and Central America. So here they are. The first systematic rule is that bird-pollinated species tend to be, if anything, more red or orange than bee-pollinated relatives. And we actually have some kind of mechanistic science that would lead us to expect that. That is, if you look at the pigments in the eyes of bees and birds, then bees are not particularly good at telling the difference between red and orange. They have pigments that are very sensitive to blues and ultraviolets. They can tell the difference, of course, from a yellow and a blue. Whereas birds have 4 different pigments in their eyes, just like the book was talking about. And they have these little oil drops also in their eyeballs, so that refracts the light. They're able to see color better than we are. There's an extra dimension to color that they can see, and they're fantastic at telling reds and oranges apart and telling them from greens. So you could say we predicted this to be true, and then we could go through our different phylogenetic blocks and ask, was it actually the case? Similarly, we could ask, we could predict that bird-pollinated species would tend to produce more nectar that is more dilute and more sucrose-rich than bee-pollinated relatives, and so on and so forth. Now, most of these hypotheses were posed long before we came on the scene. This last one has to do with our pollen presentation theory, and that is that bird-pollinated species tend to have anthers. Remember, the anthers are the structures that present pollen to pollinators. Anthers that open wider or more simultaneously than bee-pollinated relatives. Okay, well, I'll get to the test of that hypothesis, but I want to give a little mental milepost here, separating this first section of the talk from the next section. So the last topic that I just talked about, it says, our theory suggests that because of conditional parasitism, once a lineage becomes reliably visited by a better pollinator like a hummingbird, then it's gone beyond an adaptive threshold, and there's been kind of a bifurcation in what is adaptive. So the bees are no longer, the features that attract the bees to the flowers are no longer adaptive. It's the features that attract the birds. And my next topic is how do bees and birds differ in their actual pollen transfer dynamics? Or is it really the case that birds are better for male function than bees? So this next little thing, we're going to count pollen grains. And what we do is we get a little flight cage here, and it's got a trap door. You put a hummingbird feeder in there. The hummingbirds come visit it, pull the trap door, then you have a hummingbird inside of a cage. You can then present a series of flowers to this hummingbird and measure how much pollen it removes from a flower, and then how much pollen it subsequently deposits on a series of other flowers. Also, you can catch a bee, put it in the refrigerator, cools down, grooms all of its pollen off, then you let it loose in the cage, and again present flowers to it, and see how it removes pollen and deposits pollen. This allows us kind of to test what you might think of as the assumptions of pollen presentation theory, or maybe in other words, to calibrate pollen presentation theory. And then after we've done that, we'll test the outcomes as well. Okay, so we predicted that hummingbirds would be better than bees because they are not really built to collect pollen. They don't have combs and brushes for moving pollen around, and they don't really groom that much. They might preen a little bit, but not very frequently. So we measure all sorts of stuff, and maybe this is the one to pay attention to. The pollen transfer efficiency of bumblebees in our cage is 1.6, and the pollen transfer efficiency of hummingbirds in our cage is also 1.6. So the overall efficiency of removal and deposition is not really different between the birds and the bees as you average it. However, for the bees, it's a very steep function. So very early on, the bees deposit a lot of pollen on stigmas, but then it flattens out. And so there's a curve there. Whereas with hummingbirds, it's not steep. It keeps on going and going and going and going. So they're spreading the pollen out across many stigmas, and we ran it for 15 flowers, but I think they would keep on going. Like if you get to the 16th or the 17th flower, 18th flower, 20th flower, the hummingbirds would still be depositing pollen, and the bees not so much. So this difference in the pollen carryover curves is consistent with our hypothesis, and we were kind of happy about that. Now, I'll have to say that these bees in the flight cage are kind of the best bees possible compared to all other bees that visit Penstemons. These bees in the flight cage, they weren't really very interested in collecting pollen because they were in a flight cage. They were cold, and so they were interested in collecting nectar, which they use as an energy source. Adult bees don't actually use pollen as an energy source. They're just collecting it for their babies, for their larvae. So whereas some of these other bees are solitary bees, or there's a wasp, and so on, and they more actively collect pollen than the bees that we had in our cage. And so I'd say, if anything, we chose the bee that would be the kind of the best competitor in the race, as judged by flowers, between hummingbirds and bees. Okay, now we can move on. That first comparison was looking at birds and bees visiting bee-adapted flowers. So we were only using, in that first experiment, the bee-adapted flowers, and then we were presenting them to bees and presenting them to birds. Now we can compare birds visiting the bee-adapted flower to visiting a bird-adapted flower. And here we find that there is a big increase in efficiency, which is the way you would expect it to be. Once the flowers are adapted to birds, then the birds are very good at moving the pollen compared to when the birds are visiting flowers that are adapted to bees. And the shape of the curve is the same, but it's much higher. It's a much higher curve, which means that the birds are moving much more pollen on the bird-adapted flowers. And the way that they do this is that the flowers stick their anthers and stigmas way out in front, so that when the bird comes in, it hits the feathers on their forehead, which is a great place to carry pollen, rather than hitting it on their beak, where it's a pretty bad place to carry pollen. So the flowers, and also the floral tube becomes narrower and guides the beak in to just the right place. Okay, so I would say that in this little section we said that the hummingbirds are surprisingly good at removing pollen of bee-adapted flowers. As supposed, they have these long pollen carryover curves and thus are good at pollen delivery. And then they also, not surprisingly, when the flowers become adapted to hummingbirds, then the flowers are better pollinated by hummingbirds than by bees. This moves us on to a prediction, and that is that the bee-pollinated species ought to have less restrictive pollen presentation. The bird-pollinated species ought to have less restrictive pollen presentation than the bee-pollinated species. In other words, the bird-pollinated species, they ought to open their anthers up and let them be pressed against the bird's head, whereas a bee-pollinated species, it should just let a little bit of pollen out at a time and then have lots of bees pick up just a tiny amount and spread it around because of that decelerating curve. So we pretended we were bees by cutting little pieces of velvet and we take the piece of velvet and rub it against the anthers every half hour, and then we would go back and melt the velvet on a slide and count the number of pollen grains under the microscope. You can imagine this is incredibly tedious, like counting thousands of pollen grains on velvet under a microscope for lots of species. And we more or less gave up eventually, but I'll show you the data for the ones we did do. So this is just 2 flowers. I'm just picking out 2 flowers, picking on 2 flowers. We found that the pollen is dispensed more gradually in a bee-pollinated species, that's Penstemon gentrii, than in a related bird-pollinated species, a closely related bird-pollination species. So this is the cumulative number of pollen grains that are released onto the velvet, and this curve keeps on going up, whereas this one, it jumped up right away, maxed out, after a few sessions. Now, we have lots and lots of runs like this, that we didn't just do 2 flowers. We didn't give up after 2. We did hundreds. And so from each run, you can calculate statistics like the half-life of pollen in anthers, or some number that represents how fast the pollen is removed from the anthers. And so now what we've done is something like independent contrasts. So remember, independent contrast is this comparative method where you take pairs of closely related species or something like that, and you make comparisons between them. And the comparison we're making between them is bee-pollinated ones versus bird-pollinated ones. So that's one of the traits, is whether the flowers are bee-pollinated or bird-pollinated. And the other trait is the half-life of pollen in the anthers. And if it matched our hypothesis, then all of these lines would slope downward. This one doesn't quite slope downward. It kind of goes up just a tiny bit. So it's sort of the exception to the rule. And we only have 4 cases here, so it's 3 out of 4. Not super strong evidence, but it is sort of in the direction that we predicted. Now the problem is it takes years of counting pollen on velvet, and you'd go blind and crazy if you tried to do 200 species of Penstemons. But we have a few species here, and so then what we did is we went and compared our measure, half-life of pollen in anthers, to just how much the anthers look like they open up. If you just take a picture of them when they're fully dried out, then you can put the species in order from the ones that open up completely to the ones that just crack open a little bit. And I'll call that the rank anther dehiscence gape among species. And it turns out that that's related to this half-life of pollen in the anthers. The half-life of pollen in the anthers, that takes a long, long time and lots of effort to measure. This takes almost no effort to measure. So we'll go with the one that takes almost no effort to measure. And then we can do a bunch of paired comparisons, not just 4 paired comparisons. So here's one. You get one that kind of looks like a tostada and has pollen all over the top. And then we compare this to one that's sort of like a pita pocket that sort of opens up a little bit. And then here's another one. Tostada, pita pocket, and then these are crazy ones, like this is a gaping mouth, raw, raw, and this one, it just opens up by a tiny little gape. It's just a pursed lips like that. And then we call those ones over there origami anthers, because they fold back in these crazy ways. Anyway, these are 2 more cases, and you can just go through lots and lots of cases and compare how much the anthers open, depending on whether they're bird-pollinated or bee-pollinated, from every phylogenetic block, every pair of species, or sometimes we have a little bit more than just 2 species. You can ask, did it follow the hypothesized pattern, or did it not? And here are all our pairs, pair A, pair B, all the way to pair R. And there's some statistics there, but then over at that other column, you can see Are the ranks as predicted with bee less than bird? And the answer is yes, 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 yes but close, yes but close, yes, 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 no but close, yes but close, hard to compare, yes, 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 no, no but close. So as predicted, the hummingbird pollinated species had anthers that opened wider than the bee pollinated species, and this is significant for 18 pairs. So then we wanted to know, what about the ties and the near ties? And our theory, this theory of pollen presentation, it was a lot more elaborate than what we're just testing. So we were testing dispensing mechanisms. But aside from dispensing mechanisms by which single anthers can meter out the pollen onto the bees, flowers may also package pollen in time in a more staggered way, like by opening up one anther and then waiting a long time, and opening up another anther and waiting a long time, and opening up another anther. So we went and studied one of these no's to see if it's really a no. And this was done by a student. So she looked at these flowers every 2 hours, and Penstemon strictus, the bee-pollinated one, it had more censuses in which new pollen was ready for removal than Penstemon barbatus, the bird-pollinated one, and that's significant. Penstemon strictus anthers were partially open 25% of the time, whereas Penstemon barbatus anthers were never seen to be partially open. So with Penstemon strictus, it's opening up one pollen partially, and then another another one, whereas the Penstemon barbatus anthers, they just basically pop open, in one very short period of time, and it's usually all the anthers at once. So this is one of our near ties, and it turns out to be a yes if we look at it more carefully. Okay, well, one thing that you might be wondering about, if you wonder about these things at all, that was a joke. If you wonder about these things at all, you might be wondering about whether the flowers also differ in the amount of pollen that they make. Like animals differ in the amount of sperm they make, so maybe flowers differ in the amount of pollen they make. And that could really make the story much more complicated than what I presented so far, because what I've been presenting so far is all just thinking, oh, they have a fixed amount of pollen, and then they deploy that pollen in different ways. So we went out and counted pollen production, and we did this for 50 species. Now, of course, flowers that are bigger make more pollen. That's not that interesting. So we had to adjust for that, and we adjusted for it in a couple of ways. One is that we included in the model, in our statistical model, the number of ovules, which is also a measure of how big the flowers are. And of course, pollen production is related to the number of ovules. We also included the size of the anthers, and it is related to that as well. But then after you adjust for all that stuff, then there's no difference between bees and birds in how much pollen the flowers produce. And so, and then we did some other analyses. In a separate bivariate analysis, pollen production was not related to the dehiscence rank of the anthers, that predictor variable that we've been using. So it seems like pollen production is not complicating our conclusion that bird-pollinated species evolved to present pollen more generously, and bee-pollinated species start out with pollen presented more stingily. You could say that presenting pollen stingily and not opening the anthers fully, that's a symplesiomorphy. And then over and over again, you have convergence onto bird pollination, and associated with that convergence onto bird pollination, the anthers present their pollen more generously. Okay, so this last little section, I've shown that this one rule I care about, is upheld, and that is that the ornithophiles, the ones that are adapted to hummingbird pollination, have less restrictive pollen presentation than the melitophiles, the ones that are adapted to bee pollination. And the next topic I want to go into is how discontinuous are these two modes of pollination. And related to that, how conservative or labile is pollination mode among the different species of penstemons. And here's some data. So we went out and measured the characters for 49 species of penstemons, these characters that have been implicated in pollination syndromes. And then you can run it through this ordination program, which is sort of like a distance measure, but it doesn't make a tree. It's instead sees how to separate different species. And you can see that you come out with two nice separate groups on the first axis of variation, the bird-pollinated species and the bee-pollinated species. So they're fairly distinct. This could be overstated. It's not, they're not absolutely distinct, but they're fairly distinct. And then I've overlaid onto the ordination here the visitation by animals, and you can see along that first axis, that horizontal axis, that's the difference between hummingbirds and these three kinds of bees. And then the second axis of variation is kind of an axis between larger bees, Xylocopa is a large bee, and smaller bees, Osmia is a smaller bee. So on that second axis, there's not so much a distinction or discreteness of the different groups. They sort of slosh into one another. Okay, moving right along, we can also look at nectar traits, comparing bee to hummingbird pollinated species. And as you can see in the first graph, hummingbird pollinated flowers produce much, much more nectar than bee pollinated flowers. And in the second graph, bee pollinated flowers produce more concentrated nectar than bird pollinated flowers. And in the third graph, you can see that hummingbird pollinated flowers have nectar that's sucrose rich relative to the amount of hexose, whereas bee pollinated flowers, it's sucrose poor relative to hexose, so it's mostly fructose. And then we can put all of this onto an evolutionary tree. So we tried to infer the evolutionary tree using ITS data. This is stuff that we think is neutral. It's spacer regions between genes that make ribosomes. And there's no reason to think that the amino acid sequence or anything like that would be changed. So it seems like it'd be totally silent. And you can see that the red, the hummingbird pollination, it arises in different places in the phylogeny, all these different little places on the phylogeny. We also sequence chloroplast DNA, and again you can see that there's all these different places on the phylogeny where it arises. And then we did an analysis to see for a bunch of bootstrapped trees, so trees that were gathered from the same data set by resampling with replacement, the number of shifts on the tree between bee pollination and hummingbird pollination. And this number of shifts is pretty high. So for most of these bootstraps, it's in the 20s for the nuclear gene, and for the chloroplast gene, it's in the upper teens. We don't have all the species. We have fewer species for chloroplast genes than for nuclear genes. But this would be kind of our minimum estimate. Like, we think that there's a lot of shifts between hummingbird and bee pollination, probably quite a bit more than these numbers represent, not less. You can also go in and ask how many more DNA steps would you have to invoke to bring together two species like Newberii and Rupicola, which both seem to be hummingbird pollinated, so that they would be a monophyletic group. And here's a bunch of numbers. And the message from these numbers is that there's quite a bit of DNA signal that separates these different closely related bird-pollinated species. And we can pick out the ones that have high numbers. And if you pick out the ones that have high numbers, then you conclude that there's at least, I can't remember what it is, maybe like 12 originations of hummingbird pollination that would require you, if they didn't occur, it would require you to invoke at least 5 changes in DNA in a convergent way that could otherwise be explained by simple homology. And so that allows us to kind of pick at each one and find out how much evidence there is for each one. Now, there's some other cases, too, that are kind of interesting where you've got several bird-pollinated species, but they're all bird-pollinated in kind of different ways, which is not the case for the bee-pollinated species. When you have closely related bee-pollinated species, they're all spitting images of one another. You can't hardly tell them apart. But these 3 bird-pollinated species that are all close relatives, they're all bird-pollinated in different ways. Like this one over there, the bottom lip curls up, and this one here, it only folds back, and then the one in the middle has tiny lips. And that, again, suggests that they're analogies rather than homologies, whereas the bee-pollinated ones seem much more homologous. And then also there's partial steps, so cases where a species or lineage seems to have gone over to bird pollination, but it hasn't entirely gotten rid of bee pollination. And then another closely related species where it's gone completely to hummingbird pollination. So that at least implies that there was 2 half steps. A step first to acquire birds, and then another step to become exclusive on birds. You know about going exclusive, right? There's that language from the past. Like if you're just seeing somebody, you can still see other people, but then at some point you say, we're going exclusive. And that means you can't see anyone else. And that's what I'm talking about here. Like once they go exclusive, then they don't attract the bees anymore. Okay, what else is going on in this genus of 280 species? Is there any other shifts to other types of pollination? And the answer is only a couple, and they're not very convincing. So this one you might think it's got white flowers, and they are kind of tubular, so you might think, oh, moths. But no, people have gone out and studied this species, and it's still pollinated more by bees, and moths are hardly important at all for it. The one case that's the most convincing is this clade that's made up of 2 species, Penstemon ambiguus and Penstemon thurberi. And both of them seem like they've kind of given up on the whole nectar thing. So most Penstemons, they attract their pollinators primarily by nectar. They make a lot of nectar and resecrete it when it's emptied. Whereas these make a tiny amount of nectar, hardly any. And they're pollinated by flies, and also pollen-collecting bees. And the flies maybe come to visit these flowers as a place to meet up with other flies. And then the pollen-collecting bees, of course, are collecting pollen. And so that's kind of the craziest other Penstemon. But it's not the case that among this radiation of 280 species, we've gotten the evolution of moth pollination and the evolution of hummingbird pollination, the evolution butterfly pollination and fly pollination and all the different types of pollination. Instead, we had lots and lots of species that were just churned out that were all bee pollinated. And then over and over again, we had the evolution of hummingbird pollination from those species. So there have been many shifts between bee and bird pollination. It could be over 20, but there's overwhelming evidence for 10. It's hard to distinguish between convergences and reversals, but there's absolutely no evidence for reversals. And there have been very few other shifts, maybe one. This leads me to this conclusion that there's some kind of constrained lability. It's labile in the sense that it's making lots of species. The genus is making lots of species. And it's also labile in the sense that hummingbird pollination is this option that's open to it, and it evolves over and over again. It's very easy. But it's constrained in the sense that it never turns around and goes backwards, and it doesn't go to other things. And this is sometimes also called speciational drive, or we've called it basins of attraction, where there's two different options, and one is higher than the other, and one is more stable than the other. So here's two basins of attraction, and we kind of think of this as bee pollination, lots and lots of species of bee pollination. Sometimes it jumps over to bird pollination. It never jumps back. It always stays there. It never goes over the hill and becomes moth pollinated. So to sum up then, here's like my big message. First, hummingbird flowers have evolved repeatedly among penstemons from bee flowers. This is a great example of convergent evolution. Second, anthers have evolved to be less restrictive, matching the efficiency of their new pollinators, and otherwise more generous in the way that they present pollen. Third, hummingbirds become more efficient pollinators than bees, and then bees become conditional parasites. Bees become ugly. And then I think the kind of more abstract and more cosmic message that I wanted to communicate to you is that in some sense, anthers, you can think about them in the same way that you think about antlers. You can think about the evolution of sexual selection and how it affects the radiation of phylogenies. And you can think about this evolution as being kind of constrained and not very active, and then all of a sudden it jerks and it shifts to a new pollination syndrome, and then again it is static. So that's my talk. Do you have any questions?